So good evening, welcome to those of you that I can see we've got names that I recognise and maybe one that I don't, so welcome if this is your first time or if you're joining us again for another Bioscience Lite. Our session this evening is on sequencing malaria, so we've got our guest speaker Juliana joining us, who's literally just finished her PhD on this topic, so will be giving us a really cool talk around her work there. I'm also joined by my colleagues Fran and Cindy here to sort of support the session, and we'll be talking about Fran will be talking a little bit more about some activities around the topic of malaria a little bit later. General housekeeping then, you'll have seen this slide if you've been to one of these events before, but all the events that we host are aimed at exploring genomics and what it means for our lives, and obviously in this context for teaching and education. We are happy for you to ask questions this evening to ourselves and to Juliana about her research, but we ask that you do so respectfully, and myself, Fran and Cindy are on the call to sort of moderate the space and make sure it's inclusive for everyone. If you want to get in touch with us this evening, there's a few different things you can do. We've got closed captions. If you would like to use them, feel free to. They also move around the screen, so they shouldn't be in the way. If you want to interact as we go, you're welcome to put cameras and microphones on and literally just chip in, but I know that often people are marking or eating or doing other things, so you can either leave us comments and messages in the chat box that I'll pick up as we go through the presentation. You can also raise your digital or literal hand if you want to get our attention. So just let us know if you need anything. Obviously I've sort of introduced who we are, and Cindy, I apologise, I forgot to add your face to this. It will be on there for next time, but it'd be great if in the chat box you could let us know what school you're joining us from, just so we can see who's joining us this evening, ideally your school name and your postcode. But I said we've got Fran, myself and Cindy on the call, and Juliana, our guest speaker. We are joining you this evening, at least theoretically, or at least for Juliana, literally from the Wellcome Sanger Institute and the Wellcome Genome Campus, that many of you will have heard of before. We're a world-leading genomics research institute, and Juliana is part of Wellcome Sanger, and we'll talk a bit more about her research. But the research we do on campus spans from human genetics, cancer, cellular genetics, to parasites and microbes, which is obviously where Juliana's research fits, to biodiversity as part of this the Darwin Tree of Life project, and all underpinned by this concept of biodata that, again, Juliana will sort of touch on in her talk as well. Before I hand over, a sort of sneak peek, I guess, of a course that we're hoping is going to come out sometime in the summer term, date TBD, called Genomics for Educators, that actually John that's on the call was part of the creation of. This course will be a free online course through FutureLearn, and the idea is that rather than teaching you genetics and genomics, it will give you strategies, techniques, and ideas for how to bring it to life in the classroom. So we've got a lovely case study from John about his work, so we're really excited for this, and we'll of course give you more information and like how to book onto it, register for it when the course goes live, which we're hoping will be soon, but yeah, at some point this summer term. So we've got a talk from Juliana. We'll follow that by a Q&A. We'll then let her go for the evening, and Fran will talk us through some malaria discussion activities. As always, we'll be recording this, so if you miss any of it, don't worry, you can catch up later. I will stop talking and I will hand over to Juliana for our talk.
Great, thank you. Okay. Right, is this the right view?
Yeah, that looks great.
Okay, great. Okay, hi, yeah. Thank you for the introduction, Em. I'm Juliana. I just finished my PhD at the Sanger. Oh, we've already gone to my next page. And yeah, I work on sequencing the malaria parasite using single-cell RNA sequencing. So that's kind of the focus of my talk today. And so I'll kind of zoom through this. I added this at the beginning of the original presentation to kind of give a bit of introduction about myself, because I think my journey to where I am now has been really varied. And I think it's sometimes, I think as a child, I would have liked to have heard, you know, that you don't have to know what you're doing from day one. So this is a bit of an introduction about me. I'm Canadian. I'm from well, I'm from Ontario, but I grew up in a very, very tiny island on the East Coast in Nova Scotia, where there's literally nothing to do. And so I was just very bored running around, and some researchers caught me and realized that I could be free labor for them. And so I did this. I literally sat in front of boxes that looked like this, where we had a decoy bird on a nest, and I had to count the number of times that another bird came along and kind of attacked it, because they were fighting each other for housing spaces, much like we are now. And I didn't even consider that I was going to do a career in research or anything of the sort. I mean, I was kind of interested in science. I was a kid, I think I was 11 or 12. But it was really interesting, and I got to go to Dalhousie University to see some of the results, and it was really, really fun. So that, I guess, could be my origin story of how I got into research, but really I was just super bored as a child. After that, I did decide to go to university. That wasn't in my plan, but I did end up going to university, and I studied evolutionary biology largely because of my interactions with the birds as a kid. But then I kind of decided that evolutionary biology is very difficult to study sometimes in species that are big, because you have to just sit there and wait for them to do things, and you have to wait for them to evolve, and it takes a really long time. So I got more interested in parasites and pathogens, and bacteria and viruses and stuff, because they evolve so quickly that you can really, as we know because of COVID, you can really track their variants and look at their evolution at a kind of a normal time scale that was more reasonable to me. So to do that, I went over the pond to my geographically not very accurate representation of Europe. Currently all of Canada is more north than Europe. And I went to UCL to do my master's there. And then I went, as we know, to Cambridge to do my PhD. My master's in virology, and now my PhD is in malaria. So I really have moved around in the pathogen space. I think there's a lot of transferable skills looking on any pathogen. I mean, once you know what a variant is, you can look at a variant in anything, really. So yeah, I kind of got stuck on malaria because it was an interesting evolutionary question, because you can look at the evolution of the parasite itself, but then you can also look at its vector, so the mosquito. So I found that a really cool system to study. Oh yeah, this is gonna be a whole bunch of question marks coming up, which is just highlighting that I didn't know what I was doing pretty much the entire step of the way. Right, there's me in the lab. Okay, so super brief introduction of malaria, because I think this has been done to death, but needs to be done if we're going to be talking about this today. So malaria is transmitted by an infectious bite from an Anopheles mosquito. That infectious bite carries sporozoites. Sporozoites are going to enter your bloodstream via that bite in the spit of the mosquito. And those sporozoites are going to travel through your bloodstream and head to the liver. And within the liver, they spend a week or two there replicating and kind of growing and growing and growing in the liver cells. And then they burst out of the hepatocytes liver cells and continue that cycle. And eventually they burst out of the hepatocytes and they re-enter the bloodstream. So this is kind of the main stage of the life cycle for the malaria parasite. So each one of those is going to infect a red blood cell. Inside that red blood cell, that ring stage parasite is then going to grow. It's going to kind of get bigger, use a lot of energy from the cell, and then it also replicates itself and then bursts the red blood cell. And each one of those replicants are then going to infect a new red blood cell. And that's how malaria propagates. And that's how it grows really, really fast, really, really quickly. And it's the kind of synchronized sequential bursting of red blood cells that causes most of the pathogenesis or symptoms of malaria, because as you can imagine, having 2% of your red blood cells suddenly burst all of a sudden isn't very good for you. From there, in order to transmit, they actually have to leave this asexual cycle. Sorry, yeah, this is called the asexual replication cycle. So in order to transmit, it has to leave this cycle. And when it does that, it's called gametocytogenesis. So a certain subset of these cells will decide to not continue the cycle. They'll decide to go down this route to become gametocytes. And these are differentiated into male and female. They're like male and female gametes in the humans. And so a male and female gametocyte pair need to be picked up by a mosquito, and then they mate within the mosquito, and then that continues the cycle and creates those sporozoites. So gametocytogenesis is a very important part of this process because it represents how the parasite moves between people rather than replicating within an individual host. So because of that, they're a super popular area of drug target interventions, or just any interventions really, and they're called transmission blocking interventions.
Can I jump
so
in
these
with
are
a quick
ones.
question, Julie?
Yeah.
Yeah, we had a question in the chat from John saying, is the bursting triggered by anything other than volumetric stress?
Oh, they tend to be synchronized. So within a patient, they will all burst at the same time. Is it more than just having a ton of them within a cell and then bursting? I don't know. I'm imagining yes, but I can't think of anything in particular what would trigger that bursting, because the number of they're called schizonts, each individual replicate. Well, I guess no, each individual replicate is called a merozoite. The whole thing is called the schizont stage, and the number of schizonts in there can be quite variable. So I imagine yes, it's triggered by something else. Otherwise, it would be a set number probably.
Awesome. Yeah, and then he followed up saying it's really cool that they're synchronized. So
it is very cool. Yeah. And they do, yeah, no, it is. It's kind of what causes the very classical symptom of malaria is the synchronized bursting.
Thank you.
Right. Thanks for the question. Yeah, so the transmission blocking interventions are aimed to stop transmission, as the name would suggest. A number of these exist already, and the most famous of which is bed nets. That stops mosquitoes from biting people, of course, and that stops the mosquito, the transmission side of things. But a lot of drugs are also made to target this asexual replication cycle, because that's what causes most of the symptoms. But what I work on is specifically transmission blocking interventions. So those specifically, I'm looking for drugs that are going to target this cycle. So this process of parasites leaving this asexual cycle and developing into gametocytes. And when we asked this question to the student session, we got a ton of really interesting kind of strategies you can do. And honestly, some of them I hadn't even heard of. And I was like, I need to write that down. It was crazy. Right. So the way that I do this is by using RNA sequencing. And the reason I want to do that is because I want to know biologically what's happening while these cells are leaving the cycle and heading into this kind of new stage. Obviously, they're going to be using different subsets of genes, and I would like to know what subsets of genes they're using. So to do that, I sequence the RNA. Just as a refresher, which I think is going to be truly a refresher. It's just more a refresher for the students. But the way that we can pick this up is, of course, DNA is subsetted mostly into genes, and each gene is a unit of DNA, and then that gets transcribed from DNA into RNA or mRNA transcription, and then obviously read into proteins. And so I step in after the transcription process. And so, of course, the structure of an mRNA molecule is such that we have the single-stranded copy of the gene, and then we also have this 5' cap and the 3' poly A tail. And RNA sequencing specifically takes advantage of this poly A tail that should be tacked on to most mRNAs and some non-coding RNAs. And we design basically capture sequences that are poly T. So they take advantage of that and they capture the poly A tail, and that's how we fish RNA molecules out of the sea of everything else. And then attached to those poly T tails are going to be sequencing primers to start the rest of the reaction. So that's how we do RNA sequencing, kind of in a nutshell. And so what we're looking for in each case is the relative number of one gene or another compared to the total number of genes we have in the pool. And that tells us information about the gene expression, which is the detection of that transcript within our sample. So if this sample, for example, is a whole bunch of gametocytes, and I'm seeing an enrichment for gene D and gene E, that's probably telling me that the sample is full of gametocytes. If we know that genes D and E are specific to male gametocytes, or on the inverse, if I knew that this was a sample of gametocytes and I don't know what genes are expressed in it, this would tell me that genes D and E are quite important. And that's what I have represented here. So this is something that is quite often used to show gene expression data. It's a heat map. The color red is proportional to the, this is just a toy example, but the color red is usually proportional to the number of those transcripts that we've picked up in that sample. So along the, I guess this is a Y here, we've got genes, and then along the X, we've got, for example, samples. This is in a kind of bulk RNA sequencing approach where you aren't sequencing individual cells at this point, you're just sequencing an entire sample. So it could be that I went to a patient and I got a sample of their blood, and each sample is a patient, or I've specifically subsetted them into stages myself, and each sample is a stage. And so in this case, we can start to look at genes that are stage specific. So they're only expressed in one sample or potentially in one stage. And one of the questions I asked is what, based on the genes that it's expressing, what is likely in samples 5 and 6 here? And it looks like they're expressing genes A and B in our toy example. And then what I've given as my example here is that genes A and B are specific to the ring stages. So that's how we kind of use the gene expression data to tell us a little bit about what's in the sample, if we already know some markers. And if we don't, and I know, for example, it's definitely all ring stages, then this gives us a lot of information about what's being expressed at the ring stages. Right, that's essentially what I've summarized here. RNA sequencing is a tool that allows us to investigate what genes are important during each life cycle stage by counting how many of them we're able to capture in sequence. So that's all great. Obviously, I would set up an experiment where I'd look for gametocyte specifics, stages, capture them, sequence them, and then boom, I can get information about the genes that they're expressing. But the problem with gametocytes is that they're quite rare. And that's because the asexual cycle is super efficient. You can take one individual merozoite infecting a cell that develops, grows, bursts, and they can infect, exponentially more cells, and then that grows very, very quickly. But every single time they leave the cycle, they're going to commit to gametocytes, which take 2 weeks to develop versus the 48 hours that this cycle takes, and they can't go back. And so it's kind of taking that replication power of one and making it just one. So it will differentiate to either a male or a female rather than 8 or however many, 10, 15, however many chizons could be made from that one individual merozoite. So because of that, because it takes so long, it doesn't happen that often. They're also just overwhelmed by numbers. And if they don't then get picked up by a mosquito, it was a wasted kind of investment. They can't go back to replicating within the red blood cells. I think, yeah, I have forgot about my animations. So every single time, yeah, this goes around the cycle, you get exponentially more infected red blood cells. But then if you go around the cycle and commit to gametocyte genesis, you only get one. In this case, it's highlighting 2, but it either becomes a male or a female. So it means that when I take a blood sample from somebody, or even from one of my cultures where I grow malaria in the lab, over 99% of them, for the most part, are going to be this asexual stage, and only 1% or less is going to be the gametocyte stages. So because of that, if I was to sequence everything that's going on in a patient, I'd probably miss the gametocytes. And I've highlighted that here. If these are mRNA molecules I've taken from a sample of blood from a patient who has malaria, there's really probably only going to be, in this case, maybe one of these, I didn't count how many are here, that are going to be gametocyte mRNA. The rest of it's all going to be expression from the asexuals. And so that's a downfall of using bulk RNA sequencing on these samples, is that, if I was to look at this and look at the expression, it would pretty much average out, and the averaging would lose most of that unique expression that's coming from one stage. So the really rare stages of gametocytes. So instead, we use single cell RNA sequencing. And I'll get to the advantage of that in a second, but to explain what single cell RNA sequencing is and how it works, for any who are unfamiliar, this is specifically, there's a few different ways of capturing single cells. This is specifically 10x genomics, which I think at the moment is the most popular, but potentially the people who make the other versions might not like me for saying that. But it's the one I used, so I'll describe it. Essentially, it's a kind of complicated process, but they pipe they have channels that are small enough that only an individual cell can pass through. And these cells are basically coming through in a solution that contains everything you need to do RNA sequencing, all of the reagents and everything already in that liquid. And as they pass along, they get a bead attached to them. And those beads have individual barcodes to tell you what cell you're on. I'll explain that in a second. And then it passes through this oil layer. And because most of the stuff in the solution is water-based, it creates this bubble because it's in a hydrophobic environment. And that bubble, the oil droplet, encapsulates the cell, the solution, and the bead. And so you end up with this, it looks like when you have soap and water, you end up with a whole bunch of little tiny oil droplets inside your tube, which is quite fun to look at. The advantage of this, of course, is that when we average things out with bulk RNA sequencing, all I can tell you from this is that there's a bowl of fruit, but if you use single cell RNA sequencing, you can actually break it down into its constituent parts. And as we can imagine, this is super useful for malaria and for gametocytes because they're so rare. So it means I might get 99 asexuals and only one gametocyte in a sample, but I have been able to label that is one gametocyte, and I'm able to look at just the genes that are expressed in that one cell. And I got a lot more information than when I average it all out. So this is a video describing what I just, or showing what I just described with the oil droplets and the cells. So this is the formation of the oil droplets. This big block that comes through is a bead. And then it's really quick and kind of hard to see, but what comes through this channel, a little circle, is an individual cell. And so you can see in some of these drops, we get that bead with the solution with the cell all in one droplet. And that happens right around here. So actually, I'll just highlight a little bit more about what's on the bead. The bead has those poly-T, like I talked about before, to capture mRNA. And then we'll also have a unique barcode before it starts sequencing, or sorry, after it starts the primer for sequencing. So that unique barcode will tell you that all of the mRNA transcripts from this cell that you've burst within this oil droplet, and I've then attached the poly-T on the bead. All of those, when you sequence them, will have the same barcode. And so even if you break up the oil droplets later, the thing that you've sequenced will still contain that cell barcode, and that cell barcode is how I tell that that came from that cell.
Just on your little video, someone's asked, do some of the droplets not get a cell or not get a bead? Like, is there an element of, I guess, redundancy in there?
Yes, 100%. And that's one of the hardest parts of my single cell RNA sequencing analysis, actually, is trying to find empty droplets. And trying to find, so often what happens actually is the system isn't perfect, and you can have a cell that, because it's gone through mechanical stress or whatever, has lysed on its own, and so it's spilt its transcripts, and now you've got mRNA floating around in the environment. And so if you were to encapsulate a bead in there but not a cell, you might have accidentally captured some of that mRNA, and so it still attaches and it still gets a cell barcode. But we have to have basically algorithms that try to give a kind of measure of if you only have like 50 transcripts in there, that's probably, most, if we look at distribution, most of them have, somewhere around 100 to 150, maybe like 200. So 50 is kind of suspicious. And so it gets flagged as being likely ambient RNA, junk RNA from the environment. But yeah, when you work on malaria and there's a whole bunch of different stages and they all vary a lot in how many genes they express, something that is a really good cell for one stage will look like, or sorry, a really bad cell for one stage will look like a really good cell for another stage. So it's really hard to draw the line of like what do I call a real cell that's really been captured and not just randomly environmental RNA.
Awesome.
Yeah. And a lot of droplets are empty entirely. They're just oil, but that's okay because they don't have a barcode, so they don't get sequenced.
Yeah, I imagine they just sort of, easier to ignore than ones that have got that like ambient RNA that's gonna, trick you into thinking it's a cell.
Yeah, it's when there's a bead that things get dangerous. Okay, cool. So that that is essentially the use of this tactic. We've got the now we've got the same sample I've taken from people, but we've been able to tag each cell type. So these are rings, trophozoites, the kind of growing stage, and then the schizonts, the one with the lots of little merozoites ready to burst. And they're tagged specifically with what cell they came from, so we can keep track of the genes that are expressed from that cell. And so that means when I make my gene expression matrix, so this heat map of the genes that are being expressed in each sample now gets replaced with cell, specifically with cell barcode. So, sorry, in this case, I can say instead of, in this sample, it looks like they're expressing genes D and E, I can say specifically in this cell, it looks like they're expressing stage D and E, genes D and E. And more importantly, I can capture, even if it was very few, even if it was only a few hundred out of thousands, I still captured individual cells that look like they're expressing things that tell me that they're gametocytes. And so I can zone in on gametocyte specific genes, which is very, very useful and much harder to do with bulk RNA sequencing. So one of the really important things about single cell analysis, and I think a very important up and coming area of data analysis, which I've tried to explain a little bit, but I'll kind of go into a little bit more, is this idea of dimensionality reduction. I say this specifically because we're creating data that's bigger and bigger and bigger. And as you can imagine, things are getting so high dimensional that we can't plot it in 2D, we can't see past 3D. So when you've got 5,000, there's 5,300-ish genes in malaria versus 10,000 cells, you have this massive matrix of ones and zeros and counts and whatever for each gene in each cell. And it gets way too big to deal with. And you have to extract information from it somehow, which is where a lot of bioinformatics is kind of heading. This idea of how do we take big data and squish it into understandable data. And one of the ways this is done in single cell is via dimensionality reductions, so-called PCAs, UMAPs, TSNE, TSNEs. These are all different ways of compressing the information into kind of understandable chunks. Specifically, PCA, which is most easily explainable, is a principal components. And so it will take all of your information and try to take things that are really, really similar and squish them into kind of the same variable. And then it will take, oh, this looks quite similar to this, so it squishes that into another variable, and so on and so forth, to create explainable variables. So things that are, it looks like all of these cells are expressing the same thing in the same way. So let's squish that down into just one value. So in this case, it allows us to plot individual cells and how they relate to each other. So based on those principal components. So in this case, in this kind of plot, each dot is an individual cell. And essentially, every single time that they share the same expression of the same gene, they're going to get some kind of line drawn between them, like a weight added, saying that these, there's a certain probability that these two cells are related to each other because they are expressing this one gene that's in common. And then every single time that they express more genes in common, they get more lines drawn between them. And then as that happens, they get closer and closer and closer together on this plot. So that allows us to have these little clusters. So these are all probably cells that are expressing the same kinds of things because they're probably the same kind of cell. So if we were going to think of this in a human example, maybe these are a certain subset of heart cells. Maybe these are immune cells, and these are like T cells, and T cells have been recruited to do something else, or B cells, or that kind of thing. So when you look at plots like this from single cell analysis, that's how they've been related to each other. It's all related to how similar this looks, basically, their expression matrix. And I wanted to talk about that.
Sorry, there was a question. What do the axes show? So the numbers. So you've got the minus numbers, and what do those mean?
Yeah, those are a little bit arbitrary. That depends on what kind of dimensionality reduction you've used. And principal components can be squished into negative and positive numbers, that they're a little bit hard to interpret the number themselves. It's more meant to be a kind of how do I explain this? It's more meant to be a kind of relative position to each other based on the, if we describe these as genes and we describe a principal component as a whole bunch of information from genes put together, negative might be really not expressing and positive might be really, really highly expressing. But it's not an individual gene at that point, it's a number of genes together. I hope that was okay. It's hard, it's really, really hard to put these into presentations, public engagement, because it's quite mathematical. But I think it's a super cool area of research, specifically for, I mean, I'm heading into this area in my career, and I am quite lucky that there's a lot of women in biology, but there's a hard wall when it comes to getting past this into the kind of mathematical sphere in terms of this. So I thought it was important to talk about with the students and the teachers. And there's a lot of scope for using machine learning as well to learn things from this that we can't necessarily see, learn patterns that we can't see. And that is a super cool sphere. So yeah, I think it's really, really interesting. And it's the basis of some plots I will show you. So really satisfyingly in malaria, when I make this kind of plot, it really like recapitulates the life cycle. So most single cell plots will kind of look like those individual little clusters, but mine actually, yeah, perfectly recapitulated what happens in biology. So we've got, where are my animations? So here we've got ring stage parasites in green, and you can see them coming along here. They've developed and become trophozoites. They're at this point, they're growing and eating and stealing from the cell. And then as we continue along, they replicate and become these schizonts. And this is recapitulated in my single cell data as an actual circle, which is really, really fun to see. It caused me a whole bunch of headaches, but it was very, very cool in the long run. And then really surprisingly, we can see that exact point in the single cell data as cells are leaving the asexual cycle and heading into gametocytes. So here cells are leaving, they're developing into rings, and then they leave into the gametocytes, and they differentiate into males and females in this case, which was really great because we want to target gametocytes. We want to understand what causes this switch to head from asexuals into gametocytes. I want to be able to target these at all levels. So we can see here we've got this kind of stage of development where they're sexual parasites, but they're not yet differentiated into males and females. So we cut them off. If we cut them off there, maybe we kill all the males and females, or if we cut them off here, do we kill males early on? So having cells that really capture that entire cycle all the way through was really, really exciting to see. And this is using dimensionality reduction again, where each point is an individual cell. And it took a lot of, honestly, all my lockdown, I think, was spent using different parameters trying to get this to look like this. So just to kind of finish up, what can we do with this data now that I have it? Obviously, we can look for important genes. In this case, this is actually a figure from my thesis, so I'm sorry it's not as kind of clean and compact and presentable as I would like it to be.
Amusingly, John literally just said, you said it wasn't your favorite. John's literally said that's such a cool representation of data. So,
well, thank you.
There's a boost for you.
PhD students always hate their data, so it's fine. Right, yeah. So this is just the asexual cycle at the moment. It's not the sexuals. So you can see that circle, and I've assigned a value to the cells based on kind of where they are in that circle. And then I've plotted their expression over those values. So it's called pseudotime. It's kind of like time, their progression along this development. And it's kind of agnostic of stage. And so, yeah, you can see really like malaria is quite stage specific in terms of what genes is expressed. You can see things kind of turning on and then when they're needed and then turning off. And that's what's represented by this kind of yellow color. And then the blue is when it's turning off. And this is just a larger version, much, much larger version than the heat maps that I was showing earlier in the presentation, where each of these lines, of these columns is going to be an individual cell. So that's really interesting and useful because we can start to say if this was for the sexuals, for example, which does look cool, it just doesn't look this pretty, so I didn't put it in. yeah, at any point that I'm interested in, the branching point, the early, the mid, the late, the male, female, we can kind of cut out the area that we're interested in and get the exact genes that are expressed in that area. And then what we can do with this is not my data, it's another PhD student in the group, but he did something similar, just in a different malaria parasite. He did it in the rodent malaria parasite. But he took that kind of approach. He took the genes that were interesting and then he started knocking them out. And here he's got just the branch of the males and females, and he knocked out the males. It shows it's best. He's knocked out the males specific genes. And then in every single one of these 4 trials, no males developed at all because he's knocked out this male specific gene. So that was a really good indication that that could be a good drug target, because if you target it and you disrupt its expression, you get no males at all. And same thing with the females. A few females were made, but it's a lot fewer than normal if you were to knock those out. So that's one thing we can do with this data once we have it, is start to knock it out and then design drugs against it. And another thing that we're currently doing is to take this from its kind of in vitro experimental thing into in vivo studies. So we have recently, this December, went down to Mali in Africa, and we brought the 10x machine with us, and we set up the first 10x machine, so single cell sequencer in, I think, West Africa, definitely, and potentially all of Africa outside South Africa, I think, but definitely West Africa, which was very exciting because now we can take actual clinical infections and start to investigate what genes they're expressing in one, and whether or not drug targets are working on them and how that disrupts what genes they're expressing, and that sort of thing. So that's a picture of us in Mali doing the experiments there.
Just 2 questions on this slide. I know that we're also back to the Q&A anyway. But Eleanor asked for the blue diagram with the sort of yellow peaks going through the middle. Did you have to order the genes in order to make it into that sort of diagonal line, or are the genes in like a relevant order, or yeah?
Yes. I have an animation that's getting in the way. Great. Okay. So yes, I did have to order them. I ordered the cells according to the pseudotime value. So pseudotime, again, is kind of an arbitrary value that just orders the cells based on where they are in a developmental cycle. So it doesn't always correlate with clock time, because a cycle or a process could take a really long time. I'm just counting the cells as they are existing along that process, which is how we've got the order along here. That kind of scale of orange is ordering the cells. And then to order the genes, I use something essentially that looks for the density of where expression is the highest. So you've got a whole matrix of numbers, and in each row, which is in each gene, it looks for where the highest number is in pseudotime in the order of these cells. And then it will computationally rank those for me based on the density, the position of the density. So if it says the density of this gene is like really early on and like 0 in pseudotime, whereas the density of this gene is maybe like 10 in pseudotime, it will place them quite far away from each other. So that's how I rank them.
Awesome. So I guess the question was, did you have to order them to make the diagonal lines? So I guess it's just that algorithm is trying to give you this data like this, so you can actually see this change over time.
Yeah, because this pattern does exist. I didn't just
make it look pretty.
Yeah, I did and I didn't. The pattern does exist, but if I plot the genes in random order, then it's all over the place. It looks completely unpatterned. So I knew that there is some kind of association with time, because people have showed that there is an association with time in the asexual cycle. So yeah, I kind of had the bias there of knowing that there is a reason, and so then I ordered them.
Awesome. And then for your knockout selection, John asked what was used for the knockouts.
So that's a good question. I should have read that paper before the session. I think it's the same structure as the plasma gem, if you're familiar with that. Actually, Cindy, you might be more familiar with that because you worked on plasma gem. It's the whole plasma gem framework. I don't think it could be CRISPR. I'm a bioinformatician. I'm the wrong person to ask.
No, that's fine.
I think Gareth was probably the best one to ask about it because he was doing all of that in the barcoding, but I'm sure we could share the journal after in its learning as a result.
Yeah,
Yeah, I can get the paper.
awesome. Yeah, and I think you were just about to get to the end, so I'll let you share your final slide before we sort of move into the.
Oh, the final slide is just thank you for listening. And yeah,
you got another
lovely
little mosquito.
Yeah, I do.
Awesome. No, that was really cool. And there was a couple of questions that I sort of deliberately saved for the end because they were a bit more sort of general. So John had a great question about when you were talking about when scientists sort of first worked out the sort of sexual and asexual cycle of this. Was it accepted in the scientific community immediately, the sort of finding of the sexual cycle? And he put in brackets thinking about that it must have been difficult to reproduce the data. So whether that impacted whether it was sort of accepted initially. I don't know if you know anything about that, but I thought it was a very cool question.
I do. I have some fun facts to answer that question. So the reason falciparum is named Plasmodium falciparum is because of the shape of the gametocyte. So it was actually found first. The gametocyte is shaped like a little banana, which they called falciform in old-timey Latin. So that's why they called it Plasmodium falciparum. There's no other species of malaria that we know of that have a similar shaped gametocyte or sexual stage. So it was actually found first. That's how they found malaria, is these gametocytes. And the reason for that is because not all of these stages are circulating in the bloodstream at all times. Oftentimes you're only going to get, the rings will hide away. So you might only get some of the asexual cycle, and you're only going to get the very, very last stages of the sexual cycle. So that the sexual cycle, when it happens, or the sexual development, when it happens in the bone marrow. And they only leave the bone marrow, which is kind of safe from the immune system. And they only leave the bone marrow when they're ready to be picked up by a mosquito and transmitted. So because of that, if you're going to look in the blood, you would, and because the shape is so distinctive, you would be able to see those, and that's how they found them. Some patients are expressing quite a few gametocytes, but it's really at a very specific time. Gametocytes are often synchronized with mosquito patterns. So if you have, if you're in an area where mosquitoes bite during the day, you'll tend to get gametocyte release during the day. If you have areas where mosquitoes tend to bite at night, you'll have gametocyte release at night. So because it's so rare and fine-tuned, you can find patients that have them. And so you definitely can find them, but it's kind of about right time, right place in order to find them. But yeah, the gametocytes are very well studied, I would say, for what we can do. In terms of the gene expression, people have tried to, you can do things to isolate them as best as you can. But if I wanted a kind of a pure population, it is best to use single cell, and also they don't have a sex chromosome. So the only way to tell the difference between a male and a female, or even a male/female from the asexual cycle, is to use the RNA expression. So it allowed us to get quite pure populations of males, females, and early gametocytes.
Awesome. We've got another question just in, but I'll say for anyone else, if you've got questions, stick them in the chat box, or unmute and just sort of ask away. But John's asked, how is cell entry mediated?
Oof.
Which I know might be slightly different from your focus, but yeah.
No, that's a great question. There are a lot of cell surface markers on the surface of mostly the red blood cell. So the parasite lives within the red blood cell and also transports its own surface markers to the red blood cell membrane as well. And a lot of these are some of the most, of course, because they're interacting with the immune system, they're some of the most variable areas in falciparum, and actually all of malaria. There's a few of them: Vargenes, Riffins, Devors. They're these large multi-gene families that are transported to the surface. It is a huge area of research looking at these things that are transported, that mediate cell entry and leaving the cell, and then a new merozoite coming and entering a cell. Interestingly, a lot of these are completely different in gametocytes. So they have their own sneaky ways of getting into cells and developing in cells and evading the immune system. But yeah, they're good targets for vaccines because they're at the surface, but they're bad targets for vaccines because they're so variable. So yeah, it's a very cool area of research that unfortunately I don't work on, but.
Just on that vaccine note, someone has asked, and I know this isn't what you've researched, but you probably know more than we do, saying what developments towards malaria vaccines are there. So I don't know if you know more than me, or Fanos, and if you know more than me on the sort of latest on that front.
Yeah, I mean, I'll say what I do know. I mean, the latest one is, or I guess maybe the most developed one is the RTSS. I forget exactly what that's targeting. Potentially CSP.
It targets the liver, doesn't it?
Doesn't, yeah.
Yeah.
Yeah, and then the CSP is another one that targets the sporozoite stages. It's called the sporozoite protein. The RTSS is showing really good promise so far. They're doing trials largely in, I think at the moment, West Africa. I met some people in Mali who are doing trials in Burkina Faso. However, the downside to it is that it needs, at the moment, as far as I know, 4 doses to be effective. And that can be quite difficult when you have A, young children, as I'm sure everybody here will appreciate, and B, it's hard to get access to healthcare facilities. If you have to then keep going back for 4 doses, it can be quite hard to get the full course of the vaccine, but it's showing really good promise. So I think it's definitely a step in the right direction. The CSP vaccine, I don't know exactly what stage that is in terms of development. But yeah, please do chip in with more, Francesca, if you know.
No, the one I knew more about was, like you said, the RTSS one, which was the liver stage. I mean, it's so hard, like you said, because they're so variable and they're constantly changing. It's is really hard with the vaccines, and most vaccines are for things like viruses, or but this is a protozoa, so I don't think there's any vaccines for protozoas apart from RTSS. So it's a really difficult, it's really interesting area, but it's also a really complex, difficult area as well. But yeah, it's really cool.
Yeah, it's very, very interesting. And especially, well, at least with drugs as well, because the stages are so different. Like, it's the same DNA, it's the same genome, but they're going through like a ton of different stages. And as you saw, being super specific with what they're expressing when because of that. And so it's almost like it's a whole other species when you're trying to target the liver stages versus the blood stages, and then the blood stages versus the gametocytes, and then the gametocytes versus the mosquito stages. So you sometimes can make a drug that stops your symptoms but doesn't stop you from transmitting to other people, and vice versa. You can create a drug that helps mediate the transmission to other people but doesn't stop you from being ill. So it's very difficult to make a vaccine and to make drugs for malaria.
Also, with that vaccine, don't they have to constantly dissect the mosquitoes to get the sporozoites to develop the vaccine?
Yeah, I can imagine. I mean, dissecting mosquitoes, as you know, is the worst.
Yeah, yeah, mosquitoes dissecting their salivary glands are quite hard. And I know Jake Baum's lab was working on the vaccine, but they had to dissect so many mosquitoes to just be able to produce a vaccine. So that also adds another challenge to having a vaccine for malaria.
Yeah, if only robots could do that too.
Someone said something about the vaccine being engineered in yeast. Not my area of expertise. I don't know. No.
I don't know.
Some homework from us all, clearly. So someone asked, do other pathogens express different genes at specific stages in a similar way? So yeah, there are other pathogens that act a bit like this?
Yes. I'd say parasites, or at least, single-celled organisms like this tend to have, if they have complicated life cycles, that comes along with complicated gene expression. So the other parasite that's related to malaria is Toxoplasma. It causes, it can be transmitted through cat poo to pregnant women, and it causes illness, specifically when it passes through to the fetus. It's one of the only parasites can pass through the placenta barrier into the fetus. That also has a very complicated life cycle, has to be picked up by cats or another kind of host, feline host, and pass through that way. And when that happens, you have to express specific stuff. I mean, schistosomyces also has similar kind of very specific gene expression at very specific times. I imagine viruses and bacteria too, but I'm less familiar with the expression in those. Especially viruses tend to have way fewer genes. But I do know that they express different ones when they're entering a cell, replicating within the cell, and leaving a cell. So yes, I think a lot of, especially if you go on the CDC and you see that like life cycle diagram in the first part of the CDC website, you just know there's going to be a whole bunch of different gene expression patterns happening there.
Awesome. And I think the last one that I've spotted in here just at the moment, I think from when you're talking about the sexual cycle and the sort of the male and female gametocytes, John's asked if they don't have sex chromosomes, how are they sexed, or do you just rely on the mRNA? Can you sex the cells in any other way?
Largely mRNA, but also morphologically, you can see there's a difference in the growth of males and females. There's a certain stage at which that happens. So at first they kind of just look like generic early gametocytes, and then eventually you can start to see differences between the males and females. In December when I had my Viva, I did remember exactly what was different between the males and females, but I don't remember now. But basically, because they have different biology, so the females basically just kind of sit around and wait for a mosquito to pick them up. And then when they're in the mosquito, they wait for a male to mate with them. And then they start the flurry of activity of developing an oocyst, which is essentially like an egg. But the males specifically have to, they divide themselves actually. So one male gametocyte will make 8 male gametes, and they have a little tail kind of like a sperm in order to swim through the mosquito gut and find females. So in order to prepare for that, they start to differ in how they look and kind of what is inside them. So they have different amounts of like granules and stuff for storage. And so you can tell the difference between males and females that way, but largely it's the genes that they're expressing. So if you were to come up with like sometimes in FACS, so cell sorting, you can use like a female specific gene and tag that fluorescently and then use that to sort the males and females. But yeah, that's often relying on the fact that they're mature males and females. When they're in that early stage or a little bit kind of earlier in development, if they're not expressing that gene yet, then you can't use that. And so that's kind of the benefit of my data, is we know what genes are happening in the middle there, so maybe we can help isolate them that way.
Awesome. I think that's all the questions we've had in, so I will give a second in case something pops up in the chat box, but otherwise thank you so much for joining us and giving your talk. Again, it's always so interesting to hear about your research, and congratulations on finishing your PhD. I hope this was a good way to finish it all up. A lovely question from Eleanor that I think is a nice way to finish up for you. So could you explain what the next steps in your research will be? So either you personally or what you hope your research might go on to do.
Yeah, no, it's a good question. We are trying to hit this kind of single cell aspect from every angle we can, which is a very Sanger way of doing it. We're doing kind of twofold. One is that we've now got some really good data from the field, from when we went down to Mali, and we got some infections from Mali. So we're going to be looking at what's the difference between a parasite that's circulating actively through a person and a parasite that has been growing in culture for 80 years, some of these. No, 30 years. They were isolated in the 80s. Oh my God, that's not 30 years ago. They were isolated in the 80s. They've been growing in the lab for a while. What's the difference between those two? And then we have other species as well. So we've captured not just falciparum, but people who are infected with Plasmodium ovale, which is another species of malaria that can infect humans, which is a lot less likely, and also Plasmodium malariae, which is another species of malaria that can infect humans. So we'll be comparing the gene expression patterns in those, also looking what happens when because sometimes people are infected with both. So we've got people who are infected with falciparum and malariae. How does that change their expression? So that's one aspect, is we're going to be kind of creating this network of gene expression in those species and in the field. And then I'm also looking at things other than gene expression. So I'm looking at non-coding expression, so that kind of dark matter of the genome. Malaria has it too, and they seem to be quite stage specific as well. There's a famous example of those proteins I talked about that get exported to the surface that are super variable. Their expression is very specific. They only express one at a time, and that likely is mediated by non-coding RNAs. So little non-coding things will come and stop and mediate the expression of coding expression. So I'm working on that. I've also been working on long read single cell RNA sequencing, which is yeah, instead of taking the transcript and chopping it up into bits and then trying to reconstruct it, I'm trying to capture the entire transcript's length in order to look at, because falciparum has such a small number of genes, but it needs to do so many things. We suspect that it's kind of copying and pasting different areas of the gene. So it might only use half of one gene to do something in one stage, and it might use the other half to do something in another stage. So I'm looking to see about that kind of multi-use of genes using long RNA sequencing. So yeah, it's a high order, but that's what we're going with next.
Awesome. Well, thank you so much for joining us this evening. I will give you back the rest of your evening, but thanks for, and there's lots of thanks in the chat box as well, so thank you very much for joining, and I can see John's virtual round of applause. I'll hand over to Fran to talk about some activities that we have relating to malaria and sort of teaching that in the classroom. So I will reshare my slides, but yeah, thank you very much, Juliana.
See you.
Thank you.
Thanks.
Brilliant. So yeah, that was absolutely fantastic as always. So yeah, I'm going to just sort of highlight a few, this probably won't take that long, but I'll just highlight a few resources that we have that can support the teaching of malaria in the classroom. I know some syllabuses do cover it, some don't always, but I think it's just a really good conversation to have about this particular disease, because it affects so many people, and it's just such cool biology. I mean, it's just like, wow. I mean, if you can't get excited about that, I don't know what you can't really, because yeah, I love it. I love everything to do with parasites and microbes anyway. Shameless plug, and like I said, a freebie alert as well. So there is this book, oh, I got blur on, here you go. Got this book, Genomics, and there's a lovely chapter on parasite genomics, which covers some of the aspects that Juliana has talked about, and including talking about those variabilities. There's one that talks about trypanosomas and how their antigens vary and are constantly varying, which is why it's so difficult to treat that particular type of protozoa as well. So if anyone hasn't got this book and would like it, give us your details in the chat or privately, and I will post one to you, as there's only a few of you on the call tonight. And for those watching asynchronously, I will also offer that to you as well. We have lots of them, so let's see how many people watch offline, shall we?
I was going to say, that's a great measure of who's watching later.
Yes, so yeah, and it's a great book, and it's got not just about parasite genomics in it, it's got about lots of other things in as well, but I just feel like this would really support what you've heard Juliana talk about today, particularly as well. Brilliant, John. I will of course post you one, and anyone else who wants one. Other resources. So I'm pretty sure if you've been on one of our calls before, you will know about the lovely website yourgenome.org. So this has got, you can see there, it's just at the bottom there, it's got lots of different content, but we do have some lovely pages on malaria. So fact pages, these are really great for students, quite accessible because they're quite sort of bullet point sort of facts and information, but we do also have stories as well, which give almost like a more magazine style article, but take a deeper dive into the subject. So this one talks about how it's the master of the site, like we told, how it's constantly changing, and how that's just why it's so difficult to find drugs and vaccines for it. I think we do also have one on the development of the RTSS vaccine as well. So there's some lovely stories specifically about that if your students do want to delve a little deeper. We do also have a couple of resources as well. So we won't show this whole video because it's 8 minutes long, but if you want your students to watch or have a very general overview of malaria and the different stages and how you can potentially prevent it, we have a video. I think Emma's going to try.
Yeah, I've managed to close the tab, which is always a good start.
It's on our website. It's on YouTube as well, of course, as everything is. But we'll show you that in a second.
For some reason it doesn't want to go to full screen, but
oh, what a lovely Trello board.
Yeah, that is you can tell I've had a long day. Apologies.
So you can see our planning of activities that we do. There we go. Yeah, I'll just literally just show you a few minutes of it.
Malaria is an infectious disease caused by a parasite called Plasmodium. It is spread from person to person by infected Anopheles mosquitoes. The parasite's life cycle is complex. When an infected mosquito feeds on a human, it injects parasites, known as sporozoites, into the blood. These parasites travel to the liver where they develop into merozoites. These are released into the blood where they invade red blood cells and multiply many times. They then burst out, destroying the red blood cells, leading to the symptoms of malaria, such as anemia and fever. Inside some red blood cells, the parasite develops into
There you go. And then you'll see the gametocytes, the little bananas. They do literally look like bananas in the cells. So it goes into that. So, and these are lovely animations. We got licensed from Drew Berry, who's an amazing sort of animator, 3D animator. But it goes into that and a few other bits as well. So, but yeah, that's available there. And then we've also to support this, we have got a couple of activities as well, and these have been knocking around a while, to be honest, and we did have a lovely flash based almost website, but because it's flash based, it's not working. We haven't quite got around to extracting the best bits of that resource into something else, but that is on our to-do list. But this one I love because it essentially is a really good discussion activity, because you're given a pot of money of £20 million, so you can get your students to work in groups, and they can then decide, well, how are we going to spend that money on preventative medicine? It could be on preventative measures or preventing, helping to eradicate malaria. So you can see a few there. There's more details on each project in a document, so it's like a paragraph or two, so they can make a judgment call on it. But it's things like having mobile rapid diagnostic test kits. It could be having affordable medicine initiatives. It could be about inside residual spraying projects for rural communities, provision of bed nets, a live attenuated vaccine. So a live attenuated vaccine is sort of essentially a bit like a smallpox vaccine. It's about putting a modified but live element of the parasite in a vaccine, and then your immune system responding. But again, that's a type of vaccine that's been in development, but I don't think it's got to as far as RTSS, for example. Then there's things like traveling clinics and so on, and of course we wouldn't not have genome sequencing in there as well. So parasite genome sequencing, as this is quite an old, we haven't got single cell sequencing, so maybe we should update it, and put that in as well, as that seems to be the way things are going. So again, it's a great opportunity for thinking about, well, how can you prevent malaria? You can't necessarily eradicate it, but what are effective mechanisms? So is it by preventing the mosquito from biting you in the first place? Is it about understanding the genome? Is it about medicine and treatment? So it really sort of is quite a really nice way of understanding that juicy sort of conversation. So we do like one of those. The second one we've got is, I would say this is definitely more, I would say, for your advanced students, for your A-level students, because this gives you scenarios. There's also slides that can go with it as well, but the idea is that you are given a group, a scenario, and it could be from Cambodia, it could be from Uganda, could be Tanzania or Brazil. So they're very different settings, so it can be different continents, so it could be different mosquito types involved, it could be different parasites involved. So in Cambodia, it could be that you've got not only Vivax, but you've also got falciparum. So Vivax is interesting because once you've had an infection, it can sort of lay dormant within your body, and then maybe a few years later it can come out again, depending on the circumstances. So that's quite an interesting one. So again, it sort of helps you dig in, it gives you key facts, it gives you ideas around what is your region, what region are you trying to think of a malaria prevention strategy, and you then sort of fill in this form that says, well, who is at risk from this area, what are the main problems, and how can we tackle it, really. So it's quite a nice group thought activity. It could be something that they could go off and do if they're doing something like a youth STEM award or anything like that. So yeah, but it's quite a nice activity. But yeah, we've got urban settings in there, we've got rural settings in there, because there's obviously different factors in an urban setting versus a rural setting. So yeah, quite a nice one to get your teeth into. So I think, is that the main, there we go. So they're the main ones, but I said there are loads of great resources out there. The CDC website, which is the Center for Disease Control in America, they have lots of lovely resources on malaria, particularly the World Health Organization also has lots of malaria, but these are particularly ones that we have, and we know have had quite good feedback from teachers before. So by all means, please do go and try them out and tell us what you think. Putting the sort of the question out to the group, are there other resources that you've used on malaria that you'd recommend or would recommend to colleagues?
Or on single cell, because that's not something that we've got, it's so new that it's not something that we've got resources on.
We've got a couple of, have we got a facts page now on single cell sequencing?
We have on RNA sequencing, which which obviously is the sort of one form of it that Juliana uses, but not on single cell. Yeah, Wellcome's big picture was always good, John said.
You should be able to access them through TES, or no, certainly the STEM Learning Library. Yeah. You should get those. Anything else? Let's have a look. Okay. So yeah, so there are the main things we thought we'd share with you. Like I said, the video you can access for your genome, or you can go to the through the YouTube. John's made a board game.
Oh, I'd love to know. Yeah, please know more about that. And Eleanor, glad that you can join us. So yeah, I would recommend sort of your genome for lots of different resources, and there's a few others that I can pop in the chat box. But also as a plug, I hope that our Genomics for Educators course will also be really perfect for you, because a large portion of the course is geared around finding resources, evaluating them, and knowing how to use them. So hopefully once that's launched, that'll be an ideal one for you. But yeah, John, please do tell us more about your board game.
He's being very mysterious about his board game.
I'm typing a very long message.
Rapidly typing. Oh, it goes through the
life cycle. Awesome.
Very good. So we have got, like I said, one of the other malaria challenge activities is a card game, but it's literally the idea is that the students get given a life part of the life stage, and they sort of become experts on that, and then move around in groups, and they then discuss, well, what is the best life stage to target interventions at? So a little bit about like what Juliana was talking about, because if you hit it at the bottlenecks where there's the least parasites to deal with, so things like the sporozoite stage or the liver stage, they're quite good bottlenecks. It's when you start getting tons and tons of the asexual, when it's in the blood stage, the red blood stage, that's when it's very, very difficult to manage. So again, you sort of look at those, or the gametocyte stage, because there's not too many gametocytes coming out. So but you need probably a bit more information on those particular parts, which unfortunately you can't see as well because the resource is in Flash. But again, we are going to try and extract the best bits from that. So I feel like I'm waffling on. So any questions about these resources, or areas where you think, you should really be doing this, resources that we would like to see? Because obviously we always like to hear from you and look at what we can develop further. We could maybe revamp John's board game.
But yeah, I popped our team email in the chat box. If you're interested in a textbook, pop us an email there. But likewise, if you have a go at any of these and think of any sort of changes, or think of anything else, yeah, John would love us to remake his board game. Then yeah, pop us an email, because we're always sort of open to, we want to make what is useful rather than just what we think is exciting. So yeah, do let us know. otherwise, I think we'll wrap up a bit earlier. I know it's half term for most people, so we'll give you a bit more of your evening back. Today, our next session for Bioscience Lite will be around cancer mutation. So the student focus talk is in 3 weeks. And so the Bioscience Lite of that will be in mid-May, I believe it's the 11th. So we've got a researcher from campus coming to talk about how he uses CRISPR in studying cancer mutations, and sort of really interesting bits around changes he's made to sort of make it the best he can. He's had some really cool videos to show last time we spoke to him, so I'm really looking forward to that talk. And as ever, that will all go out on It's Learning and various emails, so you shouldn't be able to miss it. But yeah, thank you very much for joining us, and hope you have a great rest of the Easter half term, if it's Easter half term where you are, and hopefully we'll see you all again next time.